4 deals with altered cellular tissue and biology. So what happens when normal cellular function begins to break down due to insult, injury, disease? Okay. And some of the different adaptations include atrophy, which is a decrease in cellular size, hypertrophy, an increase in cellular size, hyperplasia, an increase in cell number, dysplasia, which is deranged cellular growth, and metaplasia, which is replacement of one type of cell with another. Okay. So if we look at each, okay, we could see sort of a normal pseudostratified ciliated columnar epithelium in the diagram, right? In metaplasia, what's happening? Well, injury or irritation is causing that tissue to be replaced with another tissue type. Okay, you might, for instance, see this in something like, a precancerous change. Okay, in dysplasia, you have persistent severe injury or irritation, and you get replacement not just with a different tissue type, but with abnormal tissue. Okay, atrophy, of course, is shrinkage due to essentially disuse. Okay, and hypertrophy is an increase in the size of the cell. Best example you probably know of hypertrophy is that when you exercise, your muscles get bigger. All right. And the reason they get bigger is because they create more muscle protein, not because you get more muscles themselves. Okay. Um, the the hyperplasia, okay, is just an increase in cell number. All right. So that it. Okay. So we were talking about the different types of cellular manifestations of disease. Okay. So we have covered that. Okay. How do you tell these? How do you see them? Well, you can see them with gross observation in some cases, but often you have to do what's called a biopsy, where you have to take some of the material and put it on a microscope slide, stain it, and look at these changes and compare them to normal tissue. Okay. Cellular injury occurs if the cell is unable to maintain homeostasis. It's reversible in some cases, so the cells can recover. It's irreversible in some cases, and the cells die. And so you can see here the series of steps that take place. Right, you have a normal cell, you have stress, it tries to adapt. Okay, it either does or does not adapt. Right, you will see evidence of cell injury. Okay, if it is a reversible injury, this is going to be a mild and temporary change and will return to being a normal cell. Okay. But if it is an irreversible injury, the cell is going to die. And there's different types of cell death that we'll talk about. One is necrosis and one is apoptosis. Okay. Necrosis is cell death due to insult or injury, while apoptosis is programmed cell death and is a more orderly process. Okay. Take some examples. Hypoxic injury. You should know that hypoxic means low oxygen levels. All right. So this can be the result either of a toxin that blocks oxygen binding in the blood, or this can be due to blockage of blood supply to the area. Okay. Oxygen is life. After about 5 minutes, without oxygen, cells and tissues die. Okay. It's the single most common cause of cellular injury, results from any of these reduced delivery conditions, right? Less oxygen in the air, loss of hemoglobin or decreased efficacy of hemoglobin, decreased production of RBCs, so your crit falls, disease of the respiratory and cardiovascular system. An example would be what fluid in the lungs, for instance, which can come from a variety of disease conditions. Okay. And then poisoning of oxidative enzymes within these cells. Okay. A couple of examples we'll talk about are cyanide and carbon monoxide. There's a few others. Ischemia is the most common cause of hypoxia. Okay. Ischemia just means that there is limited blood delivery to the region. In ischemia reperfusion injury, additional injury can be caused by restoration of blood flow and oxygen. The mechanisms include oxidative stress, increased intracellular calcium, inflammation, and activation of complement. So let's take a look at these types of injuries.
Welcome back. Today's topic is going to be ischemic reperfusion injury in the heart. In our previous video about cellular changes in the myocyte during ischemia, we learned that we could reverse this ischemic state via reperfusion mechanisms like angioplasty and ultimately restore the blood flow to the heart. But under certain circumstances, the restoration of blood flow to ischemic tissues can paradoxically worsen or increase cellular injury. This is the exact opposite outcome of what we expect from reperfusion. This phenomenon is known as ischemic reperfusion injury. This so-called ischemic reperfusion injury may contribute significantly to tissue damage. Reperfusion injury occurs in three major mechanisms that account for worsening of cellular injury: free radical damage, inflammation, and influx of calcium 2+ ions. Let's talk about free radical damage. Free radicals are reactive oxygen species with an unpaired electron in the outer orbit. These oxygen-free radicals are mainly produced by damaged mitochondria through incomplete reduction of oxygen, and some of them are produced by infiltrating leukocytes. Partial reduction of oxygen in a reperfused damaged cell may yield superoxides, hydrogen peroxide, and hydroxyl free radicals. These free radicals are responsible for the pathological effects of lipid peroxidation and protein modification in the molecular level, resulting in membrane damage, protein breakdown, and DNA mutations. Reperfusion causes more cellular injury through inflammation. Reperfusion introduces more leukocytes and plasma proteins to the injured cellular area. These cells usually respond to lysing dead cells and cleaning up a mess. But the deadly combination of these leukocytes, plasma proteins, and cytokines released by the leukocytes will only contribute to more cellular injury. Let's talk about the calcium ion influx. When a muscle is stimulated or contracted, calcium ions are released from its store inside the sarcoplasmic reticulum into the cytoplasm. And when the muscle relaxes, the calcium is pumped back into the sarcoplasmic reticulum from the cell. During ischemia, when there is damage to sarcoplasmic reticulum, the calcium ion recycling is impaired. This prevents the calcium ions from moving back into the sarcoplasmic reticulum and ultimately increasing the intracellular calcium ion concentration to toxic levels. Upon reperfusion, that buildup of calcium ions will cause further cytoskeletal damage, and that would result the heart being in a state called hypercontraction. This means that area of the particular heart would get stuck in a fully contracted state without relaxing. That particular part of the heart which has been further injured through reperfusion will look dark and mottled. Reperfusion injury accounts for up to 50% of the final myocardial damage seen after a heart attack.
So when we look, right, some mechanisms of ischemia shown here, right? You have a block, right? An embolus is a traveling clot. A thrombus is a clot that has entered a vessel that's too small to let it pass, and you cut off blood supply. The result is a lack of oxygen. Okay, the cell will swell up. With reperfusion, we deliver oxygen to the damaged cell, and we generate these free radicals, and they destroy healthy molecules. And the result is that the cell can become necrotic. It can die. Okay. Let's review inflammation for a minute so that we understand this process.
Although many things can trigger inflammation, our focus here is on how microbes cause the response. The process begins with the infection itself. Microorganisms trigger inflammation when they're introduced into the body and begin to grow and produce compounds that damage host cells. Resident immune cells, called macrophages, wander into the infected area and engulf and digest the invading organisms. They help to clean up the infection and, in the process, release inflammatory mediators, chemicals that call for more help. Some of these chemicals increase vascular permeability and stimulate vasodilation. Others are small proteins called cytokines that are made and secreted by cells as a way to communicate with other cells. Here they diffuse into the vasculature and stimulate endothelial cells to express specific receptors called selectins. Selectin binds to carbohydrates on the surface of neutrophils, snagging the cells as they flow by in the bloodstream and slowing them down, such that they roll along the endothelium. Inflammatory signals trigger these rolling neutrophils to express adhesion molecules called integrins on their surface. The integrins lock onto adhesion molecules called ICAM1 and VCAM1 on endothelial cells. The tight adhesion stops the rolling, and the neutrophils begin to stretch out along the endothelial surface. Damaged tissue cells in the area of inflammation release bradykinin, a 9-amino acid polypeptide that helps loosen the tight junctions between endothelial cells. Neutrophils can now initiate extravasation, in which they squeeze through the loosened endothelial wall and into the tissues, where they can help macrophages attack the invading microbes. Bradykinin molecules also bind to other cells of the immune system, called mast cells, that are in the area, causing them to release histamine. The histamine further loosens the endothelial cell junctions, allowing more fluid and cells to move out of the capillaries. Bradykinin, when it attaches to capillary cells, induces the cells to synthesize prostaglandins. Prostaglandins stimulate nerve endings, causing pain, which draws awareness to the infected area. The 5 cardinal signs of inflammation are redness, warmth, pain, swelling, and altered function at the affected site.
Next is complement.
The complement system consists of a group of over 30 serum proteins that play an important role in both innate and adaptive immunity. Complement proteins participate in host defense processes such as inflammation, attraction of phagocytes to an infected area, enhanced effectiveness of phagocytes, and lysis of cells. Complement proteins are activated by a cascading mechanism. There are 3 pathways of complement activation: the classical pathway, the alternative pathway, and the lectin pathway. The classical and alternative pathways are demonstrated in this animation. The classical pathway requires interaction of antibodies with an antigen, usually on a cell surface. Following binding of antibodies to cell surface antigens, C1, which is composed of 3 proteins, R, Q, and S, attaches to the constant region, Fc, of adjacent antibodies through its C1q subunit. C1s then acts on C2, cleaving it into C2a and C2b. Then C1s cleaves C4 into C4a and C4b. C2b and C4b bind to the cell near the antigen-antibody complement complex, and C2a and C4a are released. This complex of C4b and C2b, called C3 convertase, has protease activity and cleaves C3 into C3a and C3b. C3b becomes bound to the cell surface, whereas C3a is released. C3b then absorbs to C4b2b to form C4b2b3b. This structure is called C5 convertase. The alternative pathway does not require the presence of antibodies. Activation begins with cleavage of C3 into C3A and C3B, either spontaneously or by a blood protein. C3b binds to foreign substances. Factor B, a blood protein, attaches to C3b and is cleaved into two fragments, BA and BB, by factor D, forming C3bBB, analogous to C4b2b. Both structures, C4b2b and C3bBB, are called C3 convertase. C3 convertase rapidly cleaves more C3 into C3A and C3B. C3 convertase is further stabilized by properdine, which allows another addition of C3b, forming C5 convertase.
Now, the take home from all that, right, is that cascade, an extracellular cascade, is going to promote healing by eliminating the pathogen, but can also produce damage in healthy tissue. Okay. The mechanisms whereby it is able to clear the pathogen. Okay, we can number them. One is opsonization. Okay. And that's coating the entire pathogen in these proteins that come from complement so that it's easier to phagocytize. Okay. The other is production of membrane attack complexes, MACs. Okay. And what that does is punches holes in the membrane so that the cytoplasm leaks out and the targeted cell is destroyed. Okay. And we also are going to tract lymphocytes. And they're going to cascade on top of all this, releasing more cytokines and generating a more vigorous response to this process. Okay. The antibodies that can attract complement are IgG and IgM. Okay. And these guys are primarily found in the blood. Okay. And what happens is they can enter the extracellular fluid and engage in battle with the enemy. Okay. Now, all these are great for clearing the pathogen, but they can also produce collateral damage. Okay. And that's because this type of response in the immune system is not directed so much at a specific strain of an organism for attack or damage, but just in general, anything foreign. All right. So what we're saying with these processes is that they're well-intentioned and they're effective, but they can also turn around and damage healthy cells as well. Okay. Now, the next topic we want to sort of cover here is the damage due to these oxygen species. Okay, so let's get out of here. And let's discuss a little bit about reactive oxygen species or free radicals.
Well, I'm going to start with oxidative stress. Oxidative stress is essentially, you need to think of this as a balance. So it's like a seat. In England, we call it a seesaw. I think we call it a teeter-totter or something like that. And we've got two kind of opposing forces. We've got, on one side, we've got the antioxidants, pro-oxidants, this side. And so normally there's kind of a bit of a balance between these two. Oxidative stress occurs when you get an imbalance. So you maybe get a decrease in the antioxidants and you get an increase in pro-oxidants. You can get an increase in pro-oxidants because pro-oxidants include free radicals. Free radicals are chemicals with an unpaired electron. Normally electrons are paired. When they're unpaired, they become highly reactive. So it would be a compound X has an electron on it, okay? And free radicals are denoted as a dot, essentially. So that's an electron, but call it a free radical because it's got a dot on it. And then there are reactive oxygen species, things like hydrogen peroxide. And then various sources of these sorts of reactive compounds can be smoking, exposure to sunlight, so the UV radiation in sunlight, pollution, diet. So really many things in the environment and our lifestyle, lifestyle can lead to these free radicals, reactive oxygen species, and increase the pro-oxidants. And on the other side, antioxidants, there's diet. And you can think of obviously vitamin C, E, and so on. But there are also enzymic antioxidants inside the body, inside the cell. And these are things like superoxide dismutase and catalase. So you have this kind of balance between the two, and obviously greater exposure to these will lead to more pro-oxidants, which, if it's not balanced out by the antioxidants, will lead to oxidative stress.
So when we're talking about the accumulation of reactive oxygen species and free radicals, okay, what happens is that they steal electrons from healthy molecules and damage them, okay? And the result is, if there's enough damage, it's going to be essentially cell death by necrosis, okay? Anoxia is complete lack of oxygen. And we'll see a decrease in ATP causing failure of the sodium-potassium pump and sodium-calcium exchange. The cell will swell. You'll see vacuolization. And of course, you've got the risk of reperfusion injury, okay? So let's take a look at these mechanisms.
Hey guys, this is the Caffeinated Method. Welcome to the channel. This video is going to be about cellular injury. So what exactly is cellular injury? Cellular injury, also known as cell damage, is defined as the effects of stress when the cell can't adapt to that specific stress. So what exactly do we mean by stress? Now, we already spoke quite a bit about stress in our previous video about cellular adaptation. If you haven't already watched that video, make sure you go and check it out. But basically, stress is anything that affects the functioning capacity of cells. Now, there are several types of stresses. Types of stresses are divided into two groups, internal and external. Examples of internal stress include genetic impairments within the cell's metabolism pathway, and examples of external stress include heat, radiation, or trauma. Now, cells like to be in homeostasis, and so they will respond to stress. However, this response depends on the type of cell, the severity of the stress, and the type of tissue that's affected. Generally, a cell would like to adapt to the stress. However, if the stress is too severe and the cell is not able to adapt, then the cell will be injured. This cellular injury can be reversible or irreversible. In this video, we'll look into hydrotic change, fatty change, hyaline degeneration as examples of reversible injury. We will also look into necrosis and apoptosis as examples of irreversible injury. So, without further ado, let's get into it. As usual, you can find a copy of the notes that I'm using down in the description box below. Just make sure you don't forget to subscribe and like the video. First, let's have a look at reversible injury, specifically cellular swelling. Now, the swelling of membrane-bound organelles is a hallmark of reversible injury, and cellular swelling, also known as hydraulic change, is the most common form of cellular injury. And this is because almost all forms of stresses will lead to the impaired production of ATP. If a cell cannot produce ATP effectively, then all the ATP-dependent ion channels will not be able to function. This means that there will be an accumulation of sodium and calcium within the cell. And this is because the ATP-dependent sodium and potassium ion channels, as well as the ATP-dependent calcium ion channels, will not be able to function. It's important to note that the lack of ATP is not the only cause of increased calcium levels. Calcium is also stored within organelles, for example, mitochondria and endoplasmic reticulum. And so, if these organelles are damaged, they will release calcium, leading to increased amounts of calcium. Now, this accumulation of intracellular ions leads to an increase in osmolarity. And so, water will accumulate within the cell through osmosis. And this is what leads to cellular swelling. There are several signs of cellular swelling that can be observed. Internal organs such as the mitochondria and endoplasmic reticulum swell. The swelling of the endoplasmic reticulum leads to ribosomal detachment and therefore reduced protein synthesis. We can also observe small vacuoles of water within the cytoplasm as well as chromatin clumping within the nucleus. Finally, when we look at the cell membrane, we observe blebbing. And yes, blebbing is a scientific word. Now, if the cell had microvilli to increase the surface area, these microvilli will be lost, and so there will be a reduction in the surface area. All in all, cellular swelling is not a good thing. But hey, it's still reversible as long as we remove the stress. Another hallmark of reversible injury is intracellular accumulations, and an excellent example is fatty change. When cells involved in fat metabolism, for example, liver cells, are damaged, they are unable to adequately metabolize fat, and so this fat accumulates within the cells. Now, there are several causes of damage to liver cells, for example, alcoholism, starvation, obesity, diabetes mellitus, liver toxins, or drugs. So mild fatty change may have no effect on cellular function, but severe fatty change can result in abnormal enlargement of cells, and this may lead to complications. For example, if liver hepatocytes enlarge, they may compress the surrounding bile ducts, and this may lead to cholestasis. Another example of reversible injury is hyaline degeneration. In connective tissue, we might have the accumulation of a homogeneous translucent material that stains intensely with acid stains. Acid stains appear bright pink on histology, and a good example of hyaline degeneration is the walls of blood vessels seen in hypertension or diabetes mellitus. It's important to note that hyaline degeneration is different from fibrillar necrosis, which is cell death, although they may appear similar in histology, i.e. a bright pink color. We will discuss fibrillar necrosis in a lot more detail in the next video about necrosis. Now, there are several different forms of cellular accumulation, but we will not be going into detail in this video, but I am going to mention a few so that you have an idea. We can have the accumulation of cholesterol, and this becomes obvious with phagocytic cells undergoing atherosclerosis. Several different types of proteins may accumulate. We might have the accumulation of iron in a disease known as hereditary hemochromatosis. We have the accumulation of mucopolysaccharides in myxoid degeneration, and we might also have the accumulation of pigments like carbon in alveolar macrophages. You get the point. Several different types of substances can accumulate within different types of cells in different conditions. Alright, we're done with reversible injury. The second type of cellular injury is irreversible injury. Irreversible injury can follow reversible injury if the stress persists, or irreversible injury can be a result of severe injury directly from normal cells. Now, as the name suggests, there's no coming back from irreversible injury, and cell death is inevitable. The real question is how. There are two main ways, necrosis and apoptosis. Both these processes are very important, and I will be diving into much more detail in separate videos for each of these processes. But basically, necrosis is a violent form of cell death that is not controlled and results in excessive inflammation, whereas apoptosis is controlled and a programmed form of cell death. In apoptosis, the cell shrinks and forms apoptotic bodies that are then engulfed by phagocytes, for example, macrophages. And this results in minimal inflammation, which is in contrast with necrosis, whereby the cell sort of explodes. Now, membrane damage is a hallmark of irreversible injury. There can be plasma membrane damage causing the release of intracellular components that leads to the excessive inflammation in necrosis. There may be mitochondrial damage that leads to the release of cytochrome C. And cytochrome C is an important player in apoptosis. And we could even have lysosomal membrane damage, causing the release of catabolic or digestive enzymes. Finally, the hallmark of cell death is the loss of nucleus. Now, there are three main processes by which cells lose their nucleuses: pycnosis, chirorrhexis, and chirolysis. Pycnosis means condensation, and so the nucleus shrinks until it disappears. In chirohexis, the nucleus is fragmented into tiny pieces, and in chirolysis, the nucleus fades away. Now, there can be several different types of necrosis, and we'll be talking about the different types in my next video, so stay tuned. That's all for cellular injury and cell death. Make sure you guys subscribe to the channel, like the video, and share this video with anyone that might find this useful. I also have an Instagram page, caffeinated.medic. Make sure you check it out for educational and medical content. Thanks for watching.
So with hydropic degeneration, we have the injury, we have hypoxia, we have a drop in ATP production. Why? Lack of oxygen delivery, okay? Remember that you make 20 times more ATP with oxygen than without it. So without adequate oxygen, without adequate ATP production, cell function begins to shut down. Sodium and water move into the cell, potassium moves out, osmotic pressure increases, more water moves into the cell. The cisternae of the ER distend and rupture and form vacuoles, and we see vacuolization. And eventually what's going to happen if this continues is that the cell is going to be completely destroyed. Okay. 5 minutes is usually the limit. 5 minutes without oxygen, irreversible damage. Okay. Free radicals. Remember that free radicals are basically oxidizing agents. And what they do is they steal electrons from healthy molecules and cause them to break down. Okay. What you'll see with these reactive oxygen species is lipid peroxidation, protein changes, changes in DNA, and changes in mitochondria. Okay. Basically oxidative stress. So if we look here at these oxidative reactive oxygen species, ROS for short, right? Normally, the way that it's dealt with, okay, is oxygen undergoes partial reduction to superoxide, then SOD changes it to hydrogen peroxide, and then there is an iron-dependent, copper-dependent reaction that produces hydroxyl radicals, and then that can lead to the production of water and free oxygen. Okay. Now, another pathway we can take at hydrogen peroxide is to see an increase in the reactive oxygen species. And so the superoxide radical and the hydrogen peroxide and the hydroxyl radicals build up. We get disruption of plasma membranes, protein destruction, and misfolding of protein and DNA mutation and damage. Okay. So how do we get rid of these reactive oxygen species? Normally is a combination of reactions starting with SOD, okay, and ending with catalase, all right, to produce water and oxygen. Glutathione peroxidase is going to convert hydroxyl radicals into hydrogen peroxide, and the mitochondria will produce from that water and oxygen. Okay, so the water and the oxygen are not reactive oxygen species. Okay, it's the partial reduction of oxygen that's the damaging event. Okay, and this is why anything that damages mitochondrial function eventually leads to the buildup of these reactive oxygen species in the cell. Okay, so you can see here, right? Xenobiotics, right? Cellular injury mechanisms from xenobiotics such as carbon tet, lead, carbon monoxide, ethanol, mercury, and street drugs. Now, let's address a few of these. Lead and mercury can be modified to form neurotoxins, okay, and kill nerve cells. This is one of the reasons that we've largely gotten rid of mercury thermometers, and we've removed lead from paint and from gasoline, okay? Carbon monoxide is a metabolic poison that sits at the end of the electron transport chain and doesn't leave. It binds 200 times more tightly than oxygen does. And so what that results in is constipation of the electron transport chain with electrons. Thus, there's no current flow. Thus, there's no proton gradient established, and the bulk of the ATP production falls off. Okay. Ethanol is can be damaging because it can increase the penetrance of other lipid soluble carcinogens into the cell. Okay. And of course, excessive ethanol is damaging because the pathway that converts ethanol into energy produces as an intermediate um, a molecule called acetaldehyde. Okay? And acetaldehyde is basically a preservative. Okay, it's closely chemically related to formaldehyde, and so people that engage in excessive consumption of ethanol can essentially pickle their liver. Okay, and all of these are going to be damaging to nerve function. Okay, chemical agents that can damage cells include over-the-counter and prescription drugs. It's a leading cause of child poisoning. Direct damage includes chemicals and drugs that injure cells by combining directly with critical molecular structures, chemotherapeutics, and drugs of abuse. Hypersensitivity reactions can result and range from mild skin rash to immune-mediated organ failure. Now, this is something that's inherited, okay? It's likely that everybody in this class has a trigger that can result in an allergic response, a hypersensitivity response. The reason everybody doesn't react the same way is because everybody's adaptive immunity is functionally identical but structurally different. And by that, I mean it does the same job, but it uses different components. Okay. So my range of defense against foreign material, which is going to be between 1 billion and 1 trillion different antigens, is different than my sister's, is different than yours. Okay. But the fact that you can defend yourself against, you know, in some cases, trillions of different antigens means that generally in a world that's filled with infectious agents, you stay relatively healthy. Okay. But you can encounter an antigen that results in an over-response of the immune system to that particular trigger. And that results in damage as a result of the immune response. Okay. So those are the byproducts of inflammation that we talked about and the resulting collateral cellular damage. Okay. A part of this is also an inherited increase in the number of basophils and eosinophils that mediate the inflammatory response. Okay. So some of the big players here are mast cells and basophils that release histamine. In the worst possible case, you can get something called anaphylactic shock, and that results in dilation of all the capillaries at the same time and a drop in blood pressure that can be lethal. Okay. So we can look at this chart here and we can see, you know, some of the chemical injury responses, right? Let's look at, in this case, they're looking at carbon tet, which is a lipid soluble compound, which means it can basically store in the body in the fat in almost indefinite amounts. Okay, that's a hallmark of any lipid soluble compound. The effect on the smooth endoplasmic reticulum, the SER, carbon tetrachloride and oxygen generate lipid radicals. Lipid peroxidation can result in destruction of the rough endoplasmic reticulum, the drop in protein synthesis, a drop in lipoprotein secretion, an increase in triglyceride content, and a fatty liver. Okay. On the right-hand side, destruction of the plasma membrane leads to permeability and an influx of sodium, water, and calcium. The cell swells, and you have a massive influx of calcium as well. This can lead to mitochondrial injury, a drop in ATP production, an influx of calcium in the mitochondria, a drop in oxidative metabolism, an increase in glycolysis, and a drop in pH. Because now, what are we doing? We're just running glycolysis and making lactic acid. Okay. This can result in lysosomal swelling, release of lysosomal enzymes, and then that's going to tear up the cell from the inside out. Okay. So all very bad in total. Okay. Intentional, unintentional, and intentional injury are more common among men and higher in African Americans. Blunt force injuries a result of application of mechanical force to the body, resulting in tearing, shearing, or crushing of tissue. Examples include motor vehicle accidents, falls, okay, assaults. You can also have contusions, lacerations, and fractures. Okay, fractures, breakage of bone, laceration is a cut, and a contusion is a bruise. Okay. Sharp force injuries, an incised wound, a stab wound, a puncture wound, a chopping wound, and there are gunshot wounds, okay, penetration wounds. Asphyxial injuries caused by a failure of the cells to receive or use oxygen include suffocation, such as choking, asphyxiation, strangulation. This is by hanging or ligature or manual strangle. One of the hallmarks in strangulation is the breakage of the hyoid bone. Okay. Chemical asphyxiants such as carbon monoxide and cyanide and hydrogen sulfide. Okay. Carbon monoxide and cyanide poison the electron transport chain. Okay. Hydrogen sulfide is a reducing agent. Okay. And then we have drowning. Okay. Infectious injury is pathogenicity of a microbe that can produce cellular damage. This includes invasion and destruction, the generation of toxins. These can either be exotoxins or endotoxins. Okay? Exotoxins are secreted. Endotoxins are part of the structure of the pathogen. Okay, and they're often released when the pathogen is destroyed, and then the effect comes after that. And they can also be, they can also result in hypersensitivity reactions, allergic responses. Okay, so let's take a break here, 20 minutes. And then we'll come back and deal with the second half. Okay, so I'll set that up. Non-living pathogens would be viruses, prions, okay, and toxins. Living pathogens, bacteria, fungi, protozoa, and helminths. Okay. How do they do their damage? Well, first of all, they get inside you, all right, generally through organ systems open to the outside world, such as digestive, respiratory, urinary, okay, reproductive, or an injury, penetrating injury like a laceration or a puncture wound. Okay, and then they can produce cellular damage through either endotoxins or exotoxins. Right, endotoxins are part of the structure of the pathogen, and exotoxins are secreted by the pathogen. Okay, now most of these are found in bacteria. Okay, there are a few in fungi and a few in protozoa. All right, most of the damage that comes from the protozoa and the helminths comes from invasion and physical destruction of tissue and blockage of structures. Okay. They can produce hypersensitivity reactions because of the foreign antigens they bear on their surface, right? And that's also true for the non-living pathogens. Okay? Phagocytic cells can eat pathogens through endocytosis and then destroy them with lysosomal action. Okay. Immune and inflammatory substances that are produced include histamine, antibodies, lymphokines, complement, and proteases. What is histamine? It's an inflammatory initiator, okay, one of several. And it causes redness, pain, swelling, and an increase in temperature where the inflammation is taking place. Antibodies are like magic bullets that are aimed against a specific molecule, an antigen. The idea is that they bind to that antigen, and then there are proteins like complement that can bind to the antibodies and destroy what the antibody is connected to. Okay. Other modes of defense include cloaking toxins, so they're not toxic, cloaking viruses, so they can't penetrate, and immunoprecipitation or agglutination. Okay, agglutination is the clumping of cells, foreign cells, or easy destruction by phagocytosis, and immunoprecipitation is the clumping of foreign material so that it can be destroyed by phagocytosis. Every antibody has at least 2 identical binding sites on the tips of the Y-shaped molecule, and that allows it to make these immune complexes that are easily chewed up. Okay, lymphokines activate lymphocytes. Okay, complement is going to it has that name because it complements the action of the immune system. Okay, and it's made by the liver. All right, and of course proteases tear up protein. Okay, so let's jump over here. Manifestations of cellular injury include infiltrations of water, lipids and carbohydrates, glycogen, and protein. What we mean by infiltration means it gets inside the cell. Okay. So let's take a look at cell injury and cell death.
Hello. Today I'm going to discuss about cell injury and cell death, specifically the mechanism, different types and causes of cell injury. In the next video, I will discuss about the cell death by necrosis and apoptosis. So, let's get started now. The ultimate goal of every cell in our body is to maintain homeostasis. However, in certain situations, due to the changes in their microenvironment, cells undergo structural and functional changes, known as cell adaptation. These microenvironmental changes could be physiological, such as increased demand and hormonal influence, or they could be pathological, such as injurious agents like ischemia and toxins. There are 4 types of cell adaptation: hypertrophy, hyperplasia, atrophy, and metaplasia. I will discuss about them in detail in another video. If the cells cannot adapt according to the changes in their microenvironment, they undergo cell injury. Cell injury could be reversible or irreversible. In reversible injury, if the injurious stimulus is removed, the cell can be brought back to its normal state. However, in irreversible injury, cell cannot be brought back to normal level, even if the injurious stimulus is removed. These cells undergo death either by necrosis or apoptosis. Causes of cell injury include hypoxia and ischemia, chemical agents, physical agents like electric shock, radiation and mechanical trauma, infectious agents like viruses and bacteria, immunological reactions like autoimmune diseases, genetic defects, nutritional imbalance and aging. Now let's discuss about the mechanism of cell injury. The first mechanism is ATP depletion and decreased ATP synthesis. Most often, ATP depletion is a result of hypoxia or toxic injury. Due to the reduced oxygen supply, the activity of the electron transport chain decreases, causing a reduction in ATP synthesis. We know that the activity of sodium-potassium pump depends on intracellular ATP. So, as the ATP level decreases, sodium-potassium pump activity also decreases. Sodium-potassium pump is essential to maintain the electrolyte balance between the cell and extracellular fluid. As the sodium-potassium pump activity reduces, consequently sodium ions tend to diffuse rapidly into the cell. Water always tend to follow sodium. Therefore, as sodium influx occurs, obligatory water absorption occurs, causing cellular swelling. Due to hypoxia, the activity of anaerobic glycolysis also increases. This causes accumulation of lactic acid inside the cell, causing reduction in intracellular pH. This results in reduced enzymatic activity. Anaerobic glycolysis tend to use up the glucose which derived from intracellular glycogen stores. As a result, depletion of glycogen stores occurs. Reduced ATP level also causes failure of calcium pumps, result in excessive calcium influx. With further ATP depletion, ribosomes start to detach from the endoplasmic reticulum, causing reduction in protein synthesis. Second mechanism is mitochondrial damage. Mitochondria can be damaged by reactive oxygen species, hypoxia, and increased cytosolic calcium levels. There are two major consequences of mitochondrial damage. One is formation of high conduction mitochondrial permeability transition pores. These channels allow calcium ions to leak out freely into the cytosol, causing loss of mitochondrial membrane potential. This impairs the activity of oxidative phosphorylation and causes further reduction in ATP synthesis. The other one is release of apoptotic proteins and enzymes, such as cytochrome C and caspases. These proteins and enzymes damage cellular components and trigger cell death by apoptosis. The third mechanism of cell injury is calcium influx and loss of calcium homeostasis. Normally, the free cytosolic calcium concentration is maintained at a very low level due to several activation effects of calcium ions. Main intracellular calcium ion stores are the endoplasmic reticulum and mitochondria. Due to ischemia and certain toxins, cytosolic calcium level can be increased. Initially due to the release of calcium from intracellular stores, followed by calcium influx through the cell membrane. As we already know, increased calcium levels can damage mitochondria and cause formation of mitochondrial permeability transition pores. Calcium acts as an activator for a large number of enzymes, including phospholipases, which cause cell membrane damage, endonucleases, which cause DNA and chromatin fragmentation, proteases, which cause protein breakdown, and ATPases, which hydrolyze ATP. Calcium ions also activate apoptotic proteins and enzymes and promote apoptosis. The fourth mechanism is accumulation of oxygen-derived free radicals, also known as oxidative stress. Free radicals contain unpaired electrons and they are highly unstable. They donate or gain electrons from cellular components while causing damage to them. Here you can see in this picture, a normal cell is exposed to free radicals, and they cause progressive cell destruction. Free radicals can be generated inside the cells by a number of reactions, including normal oxidation reduction reactions, absorption of radiant energy, inflammation, metabolism of drugs and other exogenous chemicals. Transition metals like iron and copper also have the ability to induce the generation of free radicals. Nitric oxide is produced by endothelial cells, neurons, and macrophages. Although it is usually not a free radical itself, it can be converted into highly reactive free radicals, such as peroxynitride ion, nitrate ions, and nitrogen dioxide. In normal cells, there are mechanisms to deactivate and reduce the generation of free radicals. Antioxidants either block the initiation of free radical formation or deactivate already formed free radicals. Common antioxidants include vitamin A and D, ascorbic acid, and glutathione. Metal transport proteins in the blood bind to transition metals and reduce formation of free radicals. Transferrin, ferritin, ceruloplasmin, and lactoferrin are some common transport proteins. Enzymes like catalase, superoxide dismutase, and glutathione peroxidase also play a role in reducing free radical formation. Pathological effects of free radicals include lipid peroxidation, oxidative modification of proteins by damaging the active sites of enzymes, causing structural changes and enhancing their degradation. Free radicals also cause lesions in DNA by forming cross links between the strands and causing single and double strand breaks. The next mechanism is defects in membrane permeability. This means all the types of membranes, including plasma membrane, lysosomal membrane, mitochondrial membrane, etc. Membranes can be damaged by reactive oxygen species, which cause lipid peroxidation, decreased phospholipid synthesis due to ATP depletion, increased phospholipid breakdown due to the activation of phospholipases, and cytoskeletal abnormalities due to the increased protease activity. Damage to plasma membrane results in loss of osmotic balance between the cell and the extracellular fluid, influx of fluid and ions, and leakage of cellular contents. Lysosomal membrane damage results in release of their enzymes and digestion of cellular components. Mitochondrial damage results in formation of mitochondrial permeability transition pores. Now let's discuss about reversible cell injury. There are two features of reversible injury can be identified by the light microscope. They are cellular swelling and fatty change. As we have discussed about the mechanism of cellular swelling, let's discuss about the microscopic features of a swollen cell. This is a picture from Robbins' textbook of pathology. It shows a normal cell and a swollen cell. One feature of reversible injury is increase in cell size, and cytoplasm contains fluid-filled vesicles. Another feature is distension of endoplasmic reticulum and detachment of ribosomes. You can identify it in the picture also. Mitochondrial swelling, loss of surface microvilli and formation of blebs, loosening of cellular attachments, accumulation of myelin figures. Myelin figures are phospholipid masses derived from damaged cell membranes. Ultimately, these phospholipid particles are broken down into fatty acids and glycerol. These fatty acids can be calcified due to the high concentration of calcium ions inside the cell. Clumping of chromatin due to the increased acidity is also another feature. Fatty change occurs due to the abnormal accumulation of triglycerides inside the cell. This is caused by mobilization of fat from the fat stores, which commonly occurs in the liver. Some other tissues include myocardium, skeletal muscles, and renotubular epithelial cells. These alterations in fat metabolism are commonly seen in patients with metabolic syndrome. Some other causes include toxins like alcohol, diphtheria toxin, and carbon tetrachloride, and ischemia. All right, that's all I wanted to cover in this video. Thanks for watching. In the next video, I will discuss about cell death. Hope this video made sense. If you like the video, please hit the like button and subscribe. See you guys soon in the next video.
So, infiltrations include things like pigment, okay, melanin, hemoprotein, bilirubin, calcium, which can either be dystrophic or metastatic, and urate, okay. All of these interfere with cell function. Examples, okay, bruising, extravasated red cells, phagocytosis of red cells by macrophage, accumulation of hemosiderin, and iron-free pigment, hence the coloration, okay? Calcium infiltration, right? Calcium stores in mitochondria and ER get pumped into the extracellular space and bound to calcium-binding proteins. Released after cell damage, the free calcium activates protein kinases, activates phospholipases, activates proteases, activates endonucleases. Okay, and so what do we see? We see breakdown of membrane, breakdown of cytoskeletal components, breakdown of nuclear components, and chromatin fragmentation. Necrosis is the sum of cellular changes after local cell death and the process of cellular autodigestion or autolysis. Basically, this means that the lysosome releases its enzyme payload and chews the cell up from the inside out. Okay. Types of necrosis can be coagulative, liquefactive, caseous, fat, gangrenous, and gas gangrene. Okay, so let's take a look at apoptosis versus necrosis.
When we talk about the difference between necrosis and apoptosis, there are some general themes that I think you should keep in mind when you're sitting for USMLE and COMLEX. Let's talk about some differences. Necrosis is not planned, whereas apoptosis is programmed cell death. Necrosis is always inflammatory, so it's marked by inflammation and swelling of tissue. Apoptosis, however, is not inflammatory. Necrosis is always pathological, no matter what type of necrosis it is, it's always pathological. But apoptosis is only sometimes pathological. So because it's programmed and the body's intending for certain cells to die, that's a good thing. But if that goes out of control and you have some type of mutation, which we'll get into, and develop some type of cancer, which we'll get into, that's when apoptosis can become pathological. But to summarize, necrosis is always pathological, and apoptosis is normally it's a good thing, but when it gets out of control, then it becomes pathological. Some more differences: necrosis is marked by macrophage digestion of the ultimate tissue that gets destroyed, whereas apoptosis, cells die because of a release of a pro-apoptotic factor from the mitochondria. In necrosis, the cell membrane is always destroyed, and in apoptosis, the cell membrane always stays intact, even when the cell shrinks up and dies. So these are some very broad differences. And if there's a theme that you're noticing here, it's that necrosis is noxious, and we're going to remember apoptosis. And I'll get more into that later when I talk about what apoptosis actually is and the different pathophysiologies behind them. But necrosis is noxious, which is to say that necrosis is bad. Look at the themes here. It's not planned. It's marked by inflammation, always pathological. Things are being digested by macrophages, and we're destroying cell membranes. Those are all bad and kind of extreme terms when it comes to killing tissue. But apoptosis, on the other hand, is programmed, so it's intended. It's not inflammatory. It's usually a good thing. Only sometimes is it pathological. It's pretty well controlled in terms of pro-apoptotic factors being released from the mitochondria, and the cell membranes are always intact. So even after the cells die, after you have the death and shrinkage of cells, your cell membrane still is intact. So this is apoptosis, and I'll get more into this mnemonic later, but think about Moses as a religious figure, being a good person who's very purposeful and programming and thinking about what he's doing. That's apoptosis, but necrosis is always noxious. Now I want to talk about apoptosis because it's a very complicated topic. It's got multiple pathways. There's a lot of physiology that you need to know here, and I'm going to simplify all of this for you. So the first thing we're going to do is talk about the intrinsic pathway. There's two pathways in apoptosis. There's the intrinsic pathway and the extrinsic pathway. So let's start with the intrinsic pathway. What you see here is a mitochondria, and depending on which molecule is signaling to the mitochondria, that determines whether or not we proceed through apoptosis and perform programmed cell death. So on the left side of the mitochondria, you see Bax and BAC. These are pro-apoptotic signal molecules, which is to say that when these act on the mitochondria, they promote apoptosis and lead to programmed cell death. On the other side, you see BCL2 and BCLXL, and these are anti-apoptotic factors, which is to say that if BCL2 or BCLXL are active, they inhibit the mitochondria and prevent apoptosis. So right off the bat, those are some themes that you need to understand. Now, if we're talking about Bax and BAC, what happens is these signal the mitochondria to release something from pores in the side of the mitochondria, which you see here shown as black circles. And what gets released if Bax and BAC are acting on the mitochondria and BCL2 and BCLXL are inactive, which means they cannot act on the mitochondria, then you get the release of something called cytochrome C. And when cytochrome C spills out of the mitochondria, it activates something called caspases. And caspases are the end target of apoptosis. When a caspase gets activated, it causes cell death. It kills the cell in a controlled, programmed way. So again, to summarize here, Bax and BAC activate the mitochondria to release cytochrome C through the pores in the side of the mitochondria. Cytochrome C activates caspases, and caspases kill the cell. So this is normal. This is what the body wants, because again, apoptosis is programmed cell death. So if a cell gets injured by something like radiation, if you're out on the beach too long and don't wear sunscreen, you need to kill the cells, because if the cell gets damaged, it can grow out of proportion and cause cancer. So this is why in many situations, programmed cell death is a good thing. And again, apoptosis, we think of apoptosis because it's a good thing. It's a religious figure. It's someone who's very prominent. The other reason that I use apoptosis as our mnemonic here is because Baxen-Bak, when they activate the mitochondria, they open those pores, which is to say they part the C, just like Moses did, allowing room for cytochrome C to spill out of the mitochondria, cross the river, and do its job by performing programmed cell death. So that's what happens when Baxen-Bak activate apoptosis. But what happens if BCL2 and BCL-XL are the ones that are activated? Well, in this case, what happens is you never get the release of the cytochrome C from the side of the mitochondria. And when that happens, you cannot activate cytochrome C, so you cannot activate caspases, so you do not get cell death. And instead of cell death, you get inappropriate amounts of cell survival. So if too many cells are not dying, they're living. And what happens is they grow out of control. So if you have a mutation where BCL2 becomes overactivated, which is to say that it's completely preventing programmed cell death and cells are growing out of control, that's follicular lymphoma. And you may have memorized without even realizing that follicular lymphoma is a translocation between chromosome 14 and 18, which overactivates BCL2 because BCL2 gets overactivated in that translocation. So think about it, guys. If you just brutally memorized that follicular lymphoma causes BCL2 to get translocated and therefore activated, what did that actually mean? Well, BCL2 prevents apoptosis, prevents cytochrome C, prevents caspase activation, prevents cell death, so cells grow out of control and you get cancer. You get follicular lymphoma because cells aren't dying, they are multiplying. So that's the intrinsic pathway of apoptosis. And the high yield summary is shown here. Baxen-Bak cause apoptosis by activating C and then caspases. BCL2 and BCL-XL inhibit apoptosis and never let that happen. So the end result here is activation of caspases, which causes apoptosis. That's the intrinsic pathway. Now let's talk about the extrinsic pathway. And when I'm done with the physiology on this side, we'll wrap up with some mnemonics for both of these pathways. So in the intrinsic pathway, you can have binding of one of two molecules or both to activate the extrinsic pathway. And they are shown here. Fas ligand can bind to the Fas receptor, or TNF alpha can bind to the TNF receptor. In either of these situations, whether it's one or both, you get the activation of something called initiator caspases. And these are basically like baby caspases that then go on to activate the big daddy caspase. And again, when the big daddy caspase is activated, it causes cell death. So this is the extrinsic pathway. Now, the tricky thing about the extrinsic pathway and why it's really kind of hard to memorize for USMLE and COMLEX is that there's actually two different ways that the extrinsic pathway can be activated. It can be through the Fas ligand TNF alpha binding, or it can be through the activity of a cytotoxic T cell. So if a cytotoxic T cell comes up and latches onto a membrane, it can release something called granzyme B. And granzyme B literally perforates a membrane and allows the release of something called perforin. So granzyme B breaks through this membrane and perforin enters the cell. And when perforin enters the cell, perforin goes on to activate caspases. And if you're not already seeing this theme, whenever you activate a caspase, it causes cell death, programmed cell death. That is to say, it causes apoptosis. So whether you're in the intrinsic pathway and you use cytochrome C to activate caspases, or you're in the extrinsic pathway and you bind Fas ligand to Fas or TNF alpha to the TNF receptor, where the cytotoxic T cell spits out granzyme B that perforates a membrane that activates perforin, all of these things activate caspases, which cause apoptosis. So if you're with me so far, you already know the physiology of apoptosis. You know how this works. Now let's give you some sexy mnemonics to make this easier. So when we're talking about the intrinsic pathway, our mnemonic is going to start with BCL. Now, the way that we're going to remember that BCL2 inhibits apoptosis, which causes cell death to not happen, is we write out the word BCL, as you see here in red text. And we're going to add some letters to BCL. We're going to make it into the word broccoli. So BCL is still there, we just added some letters. And what does broccoli do? keeps you alive. You live longer if you eat broccoli. Just like broccoli or BCL, BCL2 will make the cells live longer and it'll keep them alive. It inhibits apoptosis. But what about the extrinsic pathway? Where's the sexy mnemonic for that? Well, let's talk about that. So in the extrinsic pathway, I need you to remember that cytotoxic T cells release granzyme B, which perforates membranes, activates perforin, which activates caspases. I know that can seem like a lot, so how are you going to remember this? Let's write out some words that represent each step in this mechanism. So toxic for the cytotoxic T cell, granny for the granzyme B, and perf for perforin. So what happens if you're really, really toxic in front of your grandmother? You'll cause her to perf. You'll cause her to get really, really mad, maybe even have a stroke if you're not careful. So don't be toxic to granny and cause her to perf. That's the extrinsic pathway. If you're toxic, you'll make granny perf. Toxic for cytotoxic T cell, granny for granzyme B, and perf for perforin. Again, all of these pathways, intrinsic or both of the different types in extrinsic, they all end with caspases causing cell death. I really don't think it's that tough to remember, but if you're with me so far, you are a master of apoptosis, and you'll be able to answer any question that they throw at you on USMLE or Comlex. That's apoptosis. Not as bad as some of the textbooks make it sound, right? Let's move on to necrosis and quickly summarize the different types of necrosis. Now, necrosis is admittedly a lower yield topic than apoptosis, and because of that, I'm going to quickly go through this table that's going to summarize everything you need to know. There are 6 different types of necrosis that are technically fair game for boards: coagulative, liquefactive, caseous, fat necrosis, fibrinoid necrosis, and gangrenous. In each of these, I'm going to give you an example of where you'll see this type of necrosis. I'm going to tell you what you'll see on the histology, and then I'm going to give you my sexy, dirty USMLE mnemonics. Remember this. This is really, really easy and a little lower yield than apoptosis, so we're going to move through this quickly. So for coagulative necrosis, you see this in most types of ischemia. Not all types of ischemia, but like 90% of it. So if it's an ischemic event that you're seeing on your test and you have to take a guess, pick coagulative. On histology, you'll see eosinophilia. So they'll use an eosin stain and you'll see a lot of eosinophilia. And the mnemonic here is coagulation, because think about it. If you have a problem with coagulation, then that's where you have ischemia. And ischemia is coagulative necrosis, and coagulative has coag literally in the name. So problems with your coagulation cascade cause ischemia. The example of coagulative necrosis is ischemia, and then you just got to memorize eosinophilia. So I took out a lot of the difficulty in memorizing here. In liquefactive, you see this in abscesses. So abscesses classically on histology have neutrophilic debris inside of them, and the mnemonic here is liquid-factive. So instead of saying liquefactive, say liquid-factive, because if you've ever seen an abscess before, the center of it is filled with neutrophilic debris, but it's pus, right? It's liquid. So if you don't know what an abscess looks like, Jesus, Google it, because you're going to see a lot of them in your career. But abscesses are filled with liquid, pus, neutrophilic debris. That's liquid-factive, aka liquefactive necrosis. Caseous, you see in tuberculosis and in fungal infections. On histology, you see lymphocytes and macrophages forming granulomas. So the mnemonic here for caseous, instead of saying caseous, I want you to say cheesious, because fungal infections, you might say that they look a little cheesy if you've ever seen them. They have this like cheese-like quality to them. So cheesious reminds me of caseous, and I tie that back into TB and fungi because fungal infections look cheesy sometimes. And then if you know anything about tuberculosis, you already know about the granulomas on histology, so you should know that lymphocytes and macrophages are going to be present. Moving on, fat necrosis. This is in pancreatitis. It's also in trauma to the breast in women. Histology, you'll see saponification and complexing with calcium. And the mnemonic here, there really isn't one. It's just the name, fat. Around the pancreas, you have lots of fat deposition. Around female breasts, you have lots of fat deposition. So breast trauma is fat necrosis, and pancreatitis is fat necrosis. So I just remember the word fat, which reminds me of the organs that classically have a lot of fat. That's fat necrosis. The last two I'm going to group together here because they're the 2 lowest yield on this chart. Fibrinoid necrosis you see in any type of vasculitis. You see fibrin thickening of the vessel. So fibrinoid literally has fibrin in the name, so there's no mnemonic for this one. Gangrenous, you see this in distal extremity ulcers like diabetic foot ulcers. On histology, this is a combination of coagulative necrosis plus liquefactive. So you'll see a little bit of eosinophilia, you'll see a little bit of that neutrophilic debris. And the mnemonic here, also really easy, it's gangrene. So, you know, people refer to gangrene like it's a medical process. And technically, when someone says gangrene, what they actually mean to say is gangrenous necrosis. But of course, lay people don't use that term. But if you know what gangrene looks like or can imagine someone having gangrene or gangrenous necrosis on the bottom of their foot, that is the mnemonic for remembering gangrenous necrosis. But again, this chart is a summary, a little bit lower yield. Apoptosis is definitely the big topic in this video, but no coagulative liquefactive caseous and fat. And if you have the memory available, throw in fibrinoid and gangrenous. But that's it, guys. That's apoptosis versus necrosis. High yield summary of everything that you need to know and absolutely nothing more. I really hope that this was helpful.
So coagulative necrosis, kidneys, heart, and adrenal glands, resulting in protein denaturation, loss of function. You can see examples here. Okay. Liquefactive necrosis, neurons and glial cells of the brain, hydrolytic enzymes, bacterial infections such as staph, strep, and E. coli. You can see example here. Okay. Caseous necrosis, tuberculosis, pulmonary infection, combination of coagulative and liquefactive necrosis. You can see examples here. Okay. Fat necrosis in the breast, pancreas, and abdominal organs due to the action of lipases. And you can see examples here. Gangrenous necrosis, death of tissue from severe hypoxia. There's both a dry and a wet gangrene. The agent of change is Clostridium botulinum. Okay, and you can see here some examples. Various hernias where the blood supply gets cut off, become gangrenous. Okay, interception is basically the telescoping of a tubular organ. Okay, and then you can see pedal gangrene. Apoptosis is programmed cell death. It's physiological versus pathological. Self-destructive, and it's a survival mechanism. Cytoplasmic contents are delivered to lysosomes for degradation, contributing to the aging process. Okay. Aging versus disease versus your lifespan. We have to talk about the changes that take place in cells over time. Okay. Normal lifespan and life expectancy and degenerative extracellular changes. So it's not news to anybody here that we're all going to die one day. Okay. Cellular aging is characterized by atrophy, decreased function, loss of cells. Tissue and systemic aging, progressive stiffness and rigidity, and sarcopenia. The tissue function loss is linked to the cellular function loss. Okay. Frailty, we see loss of mobility, loss of balance, loss of strength, loss of motor activity, cognition, nutrition, endurance, falls, fractures, and bone density. Okay. So essentially what's going on here at the molecular level, okay, one of the big factors is loss of DNA every time a cell divides at the ends of the chromosomes that are called telomeres. And as the chromosomes shorten, more information is lost and more information is lost. And that's passed on to successive generations of cells. And eventually, when enough information is lost, the cell simply engages in apoptosis, right? It just says, that's it. I cannot anymore continue to function. And so it just hits the suicide switch and it dies. Now, the reason that we all age and die is because essentially the rate of cell death increases compared to the rate of cell regeneration. And there's simply fewer cells to do the job, which means loss of cell function, loss of tissue function, and loss of systemic function. Okay. Honestly, the faster you grow, the faster you die. Okay. And that's because of this loss of genetic material at every mitotic division. Now, there are other mechanisms as well. Okay. There's oxidative stress, there's mitochondrial damage, and so on. Okay. But essentially, once you lose the instructions that provide the information to make the tools of life, which are proteins, you can't carry on anymore. And away you go. Okay. A lot of the organisms on the planet that have long lifespans have been shown to have DNA that is basically intact. Okay. And organisms that die more rapidly, okay, have shorter lifespans, have this shortening effect as part of the process of aging. Okay. In somatic death, we have death of the entire person, and the postmortem changes include algor mortis, livor mortis, rigor mortis, and postmortem autolysis. So let's go through each of these, right? Algor mortis means cold death and describes the postmortem temperature change after someone has died, assuming room temperature, basically. Livor mortis is postmortem lividity. It's a passive process of blood accumulating within the blood vessels in the dependent parts of the body as a result of gravity, causing a discoloration of the skin that varies from pink to dark purple. Rigor mortis is that period following death where the muscles stiffen, okay, and this is due primarily to lack of ATP and calcium release in the muscle tissue, and it's then followed by decomposition, okay. Postmortem autolysis is an intrinsic activity brought about by the breakdown of cells and tissues in the human body because the constituents of the cells after death have exhibit membrane breakdown and release enzymes that begin self-digestion, basically chewing yourself up from the inside out. Okay. All right, what I want to do is